And what we're going to look at now is the next class of biological molecules, nucleotides and nucleic acids. Okay, nucleic acids are called just that because, one, they're found in the nucleus, at least most of them. There are many nucleic acids in the cytoplasm as well, but the nucleus is full of nucleic acids. The other thing, they're called acids because they form acidic solutions in water. If you actually were to take a spoonful of it, it would have an extremely sour, vinegary-like taste to it. Now, the reason they form acids in water is they have phosphate ions, negatively charged phosphate ions. Every single building block of a nucleic acid has a phosphate ion, and that makes an acidic solution. Okay, now, nucleic acids are polymers of what we call nucleotides. So let's draw a generic nucleotide. A nucleotide itself is a polymer in its own right. It consists of 3 molecules covalently bonded together. The first thing we have is a 5-carbon sugar. And that sugar is either ribose or deoxyribose, which differs from ribose just by having 1 less oxygen. Okay, ribose and deoxyribose. Now, connected to this carbon over here is going to be our base. That's a nitrogen-rich, single or double, highly polar ring-shaped molecule, single ring or double ring. And those bases include things like adenine, cytosine, guanine, especially when it's spelled, butane, thymine, and very similar to thymine is uracil. Okay, so put those 2 together. Okay, so you got that 5-carbon sugar, you got the base on one end, and down over here you have a phosphate ion, which of course has negative charges on it. There we go. So that is a nucleotide. And those are the building blocks of nucleic acids. Now the question is, how do we join nucleotides together to make a nucleic acid? Well, let's put a second nucleotide down here. So here's our sugar, here's our base, here's our other phosphate. Okay, now what we're going to do here is this. There's going to be an OH group over here. What we're going to do, or rather a hydrogen over here, what we're going to do here is we're going to do a condensation reaction and form a covalent bond from this oxygen to this carbon over in the nucleotide below. And of course we could repeat that process by having a covalent bond to the phosphate of the nucleotide above. All right. So that's the way we join nucleotides together to make nucleic acids. Now the problem is, to form this covalent bond, that happens to be an unusually endergonic reaction. Part of it is because the charges of the phosphates repel the nucleotides. So this bond formation is quite endergonic. Much more so than your typical condensation reactions. Now, in fact, it is so endergonic that the energy of hydrolyzing ATP is not enough to do that. Instead, what you have to do is take a modified nucleotide with 3 phosphates on it and pull the second and the third phosphate off. That hydrolysis reaction, remember we said we could take ATP and rip 2 phosphates off to make AMP, adenosine monophosphate? That gives you about 50% more energy. That is sufficient to do that condensation reaction. But just pulling 1 phosphate off is not. So as a matter of fact, what we actually have to use is a starting material to actually join these things together is not just the nucleotide by itself, like what I'm circling here. We have to start out with what's called a nucleotide triphosphate. Or what's often abbreviated as NTP for short. So we have, once again, the nucleotide. Now this is much like what we would see with ATP, except you can do this with others as well. You have your base, and you have, instead of one phosphate, you have 3 phosphates on here. Okay, that's what you're actually going to use to make a DNA or RNA molecule. What you do is you rip these 2 phosphates off, you hydrolyze them off. The energy released from that is sufficient to do the condensation reaction where we can join it to the nucleotide below. So that's an example of a condensation reaction that is unusually endergonic, and your typical tetraphosphate off of ATP is not sufficient, does not release enough energy to do it. So the actual building blocks, when you synthesize a DNA or RNA molecule, whether it's in the body, in a cell, or in a laboratory setting, you have to start out with these nucleotide triphosphates. Tear the 2 phosphates off, and then what's left, get the nucleotide remaining, gets joined, covalently attached to the growing nucleic acid. So that's what we see here. Okay, so that's how we join nucleotides together to make nucleic acids. And as we already discussed, different combinations of bases, hydrogen bond at different locations to others. And the combinations that are allowed are adenine forming 2 hydrogen bonds with thymine or with the very similar uracil. Cytosine and guanine form 3 hydrogen bonds with each other, so they stick together a little bit more tightly than pairs of adenine and thymine or adenine and uracil. Those are the only combinations of bases that you can get hydrogen bonds. And here again, it depends on the shape of the molecule. You have to get the bond-forming groups close enough together that bond will form. And those are the only combinations of structures that will allow that to happen. And of course, those hydrogen bonds hold the strands of DNA together, and so on. More on that kind of stuff later. All right. But before we go on to nucleic acids, we need to look at some modified nucleotides, because nucleotides are not just used as building blocks for nucleic acids. Obviously, it's a very important use, but that's not the only thing that we can use nucleotides or modified nucleotides for. Now, the first ones I'm going to mention, we already mentioned just a second ago, and that's the nucleotide triphosphates, or NTPs. NTPs are actually what are used as the building blocks for nucleic acids. And once again, you hydrolyze off the 2nd and the 3rd phosphate, and then the remaining nucleotide is covalently attached to the DNA or RNA. Now, there are a couple of nucleotide triphosphates, though, containing the sugar ribose. And those nucleotide triphosphates have additional uses. They're not just used to build up nucleic acid. So let's look at a couple of these. One of them we've already touched on, ATP adenosine triphosphate with ribose as the sugar. And of course, we know what ATP is there for. ATP is the molecule that's generally used. You hydrolyze that 3rd phosphate off, once in a while the 2nd and the 3rd. And then that provides the energy in a coupled reaction to do an endergonic reaction. So most enzymes are going to do endergonic reactions, will bind an ATP in the active site. They'll bind whatever substrate their target molecules are. They hydrolyze the ATP, and that energy release is used to power the endergonic reaction that you're trying to do in the first place. So a vast number of enzymes are going to end up hydrolyzing ATP as an energy source. But there is one other very important nucleotide triphosphate that is usually not used as an energy source. Sometimes it is, but generally this is not the case. And that's, let's replace the adenine with the base guanine to make guanosine triphosphate. Once again, this has the ribose as the sugar. Guanosine triphosphate is usually abbreviated as GTP. Now, just like ATP, you can hydrolyze 1 or even 2 phosphates off of that and release about the same amount of energy. The energetics are about the same as ATP hydrolysis. And once in a while, we do indeed use GTP instead of ATP as an energy source, but that's not normally the case. Normally, GTP has another role, and that role is in cellular regulation. Because what happens is we have a category of proteins, and we're going to go into these guys in a fair amount of detail later on, because they're so important. And these proteins are called the G proteins. G proteins have one thing in common. They bind to either GTP or to GDP. And not only that, G proteins also hydrolyze GTP. They're not very good at it, but they do. So if a G protein has a GTP attached to it, we'll hydrolyze into GDP and phosphate and kick the phosphate off. Now, what are G proteins used for? It turns out that G proteins have a different structure and different roles depending on whether they have GTP bound to it or GDP bound to it. So they have like 2 different separate states. I'll give an example here. Let's draw a G protein with GTP bound to it as a square. On the other hand, if it's got GDP bound to it instead, it's got a different structure, and we represent that as a circle in this case. And you can switch back and forth. You can hydrolyze GTP into GDP, and you can exchange this for a GTP. More details on that stuff later. But the key point is, depending on whether you have GTP or GDP bound to G proteins, they have 2 different, significantly different structural states. Most of the time, G proteins with GTP bound to them are activated. And a lot of times, the G proteins with GDP are inactive. That is not always the case, but it frequently is. So I'll put inactive in parentheses. Now, sometimes you have G proteins that do other things, do different things, even with GDP bound to it. So this active, inactive is not always true. Sometimes there are exceptions. All right. Now, when G proteins are active, what they do is bind to and regulate numerous different proteins and all kinds of different cellular processes. So G proteins and GTP regulation of them, they are involved in all kinds of things. Many G proteins are involved in signaling pathways. Signaling pathways are critically important because signaling pathways are how cells detect the outside environment, who they're sitting next to, whatever the case is, and how they respond to that. In effect, the signaling pathway is like the sense organs in your response. If I were, for instance, take an eraser and throw it at one of you folks, your sense organs would see that eraser flying towards you, and you'd respond in some way, like knock it out of the way, scream, go up to Dean and say how terrible I am, or whatever the case may be. Yes?
Can you go back a little bit? When G proteins are activated, what?
What was that again? G proteins are activated.
Negative what?
Oh, most G proteins are in the active state when they have GTP bound to them. And many are inactive when they have GDP bound to them. But that's not always the case. Some G proteins with GDP are active, but they do something other than what they do with GTP bound.
Okay, you said when they activate what?
When they're activated, what they do is they bind to other proteins and regulate. They might turn an enzyme on, for instance. Or they may bind to some other kind of protein, like a membrane protein, and cause transport across membranes or whatever. There's a whole bunch of different things that active G proteins can do. As a matter of fact, we have several hundred, if not more, G proteins in our cells, different G proteins. Each have their own different roles, okay? All right, now one of the major roles, though, of G proteins is they're involved in many, but not all signaling pathways. When we talk about cell signaling and cell signaling pathways, we will see some examples of that. Now, a second thing, G proteins, now, case in point, the importance of signaling pathways. A defect in a particular G protein involved in a particular signaling pathway, this defect where this G protein cannot hydrolyze GTP, so once it's active, it stays active permanently. Those defects in G proteins are involved in almost a third of all human cancers. By the way, they don't have a drug that inhibits that yet. I think people are working on it, but they don't have anything out of it. But, you know, clearly, that, for instance, is important in the biology of numerous different types of cancers. A G protein that is switched permanently on. Once you switch it on, there's no off switch on that guy. Okay, so that gives you one idea. Another idea, if you end up with getting cholera, right? Cholera is nasty stuff. The way cholera works is the bacteria secrete a protein called cholera toxin. That the cholera toxin happens to strongly inhibit a particular type of G protein. End result, your intestines do not suck up water, absorb water like they should, so all that water comes out along with your feces. End result is, untreated, you'll die of dehydration in hours, maybe a day or two tops. So here again, the effects of inhibiting just a single G protein. Tells you these guys are very important. Okay, now signaling pathways, one thing. Now, some G proteins are involved in protein synthesis. Some proteins that assist ribosomes in different parts of protein synthesis are G proteins. One particular family of G proteins is involved in transporting things from the nucleus to the cytoplasm or vice versa. This particular family of G proteins acts like a shuttle bus, latching on to some kind of cargo, it could be a protein or something, and carrying it from the cytoplasm across the nuclear membranes into the nucleus. And then, in the other state, it goes the opposite direction. It takes things from the nucleus into the cytoplasm. So, you know, you have your G protein bus line. Take it from the cytoplasm to the nucleus, another ticket gets you from the nucleus to the cytoplasm. Okay, so that's another kind of thing these guys do. And finally, there are many G proteins involved in regulating the cytoskeleton. That's the framework. That's a framework of protein fibers and filaments that gives cells shape, allows cells to move around, moves things around inside cells, and the like. So those are a couple of important types of modified nucleotides.